Good afternoon, everybody. I'm a PhD student in the lab of Geert Jager at the PSB department in Zwijnaarden. And I'm here today to present to you a little bit about what we do. So we are a group that focuses on the functional characterization of protein complexes through the identification of its interaction partners. And therefore, I'm totally confused. So let me start again. We are a group that focuses on the functional characterization of proteins, and we do that by identifying which interaction partners the protein makes. So the idea is that by if you know what the molecular function is of the proteins that interact with your protein of interest, you also get to know something on the function of your protein that you're studying. And so we try to map all the interactions that we find into big regulatory networks so that we can understand complex processes like growth and try to engineer them more precisely. So we have developed a platform that allows you to purify protein complexes through a setup that's called tandem affinity purification. And it allows you also to start from different kinds of plant material, like for example, we have corn and rice. And so what you do is you tag your protein of interest with a double affinity tag that consists of 2 parts, and you purify your protein to the first part of your tag. You wash away all the contaminants, and then you release the complex again and perform a second purification so that you end up with a very nice and pure complex that you can then analyze through mass spectrometry to identify the different subunits. And so my work in this group is that I've developed a new kind of tag, so a tag consisting of 2 parts, the GSyellow tag. But besides only identifying interaction partners of this of the protein, you can also identify the localization pattern of your protein thanks to the YFP that I've inserted. This is a fluorescent molecule. And you can also use this molecule to identify target genes of your protein. So that's a multifunctional Swiss knife kind of tag. And I would like to show you through 2 proteins that I've chosen how this multifunctionality is actually usable. So the first protein that I have chosen is the T-plate protein. And T-plate is a known, well, it was discovered in our department by Daniel and by Astrid, and they see that this protein accumulates at the cell plate of dividing cells and actually forms a little T at the place where the cell plate anchors into the mother cell wall. And they also saw that if you knock down this protein, you don't get this anchoring anymore and your cells cannot divide. So it was an interesting protein for cell biology. And so I fused this protein to my multifunctional tag and I performed a tandem affinity purification. And what we saw was a list of 7 interactors that are here depicted in blue. That always came up, and so we thought, what is the function of T-plate? Well, let's look to what is the function of its interaction partners, and therefore we performed on these proteins another set of interaction studies, and we found that they form interactions with proteins that are known to be involved in a very known process that's called endocytosis, and more specifically in clathrin-mediated endocytosis. And so we think that this T-plate, which apparently forms a very stable complex of 8 proteins, is present during this process where a cargo of proteins gets encapsulated in kind of vesicle and then internalized into your cell. And to test this hypothesis, we went looking to the cell wall, or actually the plasma membrane of hypocotyl cells. So these are root cells of very young seedlings. And what you see here is that the signal coming from our T-plate will form distant foci that appear and disappear every time. And you can actually measure the lifetime of these foci by a chymograph. And when we put them all together, we see that we have an average lifetime of 20 seconds. So what does this mean? Basically, this tells you that this T-plate complex comes to your plasma membrane, will help form the vesicle that will transport the cargo towards the cell, and as soon as the vesicle is budded off, so this step, the T-plate complex dissolves and you lose the signal at the plasma membrane. And so that's how we discovered the function of T-plate. Besides cell biology, we are also very interested in how development is regulated, and therefore I chose a protein that's called angustifolia 3. Now, Lisbeth did a lot of work on this protein, and she discovered that mutant plants actually have smaller leaves than normal plants. This is the normal plant. And if she overexpresses the protein, so if she makes more of this protein, you see that you have bigger leaves. So this is a very important factor in leaf growth. And so we wondered what AN3 actually does, and therefore we turned to corn, because corn is a lot bigger, and in corn leaves you have a linear system of dividing cells, followed by expanding cells, and then finally mature cells. And you can just simply dissect little centimeter pieces to have only one type of cell. And what we saw here is that there is a stable complex, like a core complex, that we always find back in both zones. And this is a SWI/SNF chromatin remodeling complex. It's a very well-known complex from animals as well. And it's known to reposition nucleosomes so that the DNA becomes open and available for gene expression so that you can start making proteins. And what we saw more, actually, is that the most interesting part, I think, is that in this division zone, there are a group of transcription factors called growth-regulating factors that are more significantly enriched in this division zone than that they are in the elongation zone. And so these DNA-binding proteins actually might have a function in maintaining the division characteristics of these cells. And to test this, we overexpressed this GRF1 protein, well, one of the GRF proteins actually. And what you see here is that the corn that overexpresses this GRF will make a lot bigger leaves. This is because you have a lot more dividing cells, or they keep dividing for a longer time. And it was a very interesting finding. And so because we know that these GRFs are binding to DNA, I thought, why not use the YFP in my tag to actually see which target genes that the protein will bind. And therefore, I performed a chromatin immunoprecipitation experiment where you cross-link your protein to the DNA. And then you cut the DNA in little pieces and you pull on your protein with a single-step purification, and you receive all enrichment of certain genes, which you can then sequence in a later step. And what I see here is that we have very specific enrichment in my sample compared to control, and that this enrichment mostly takes place in intergenic regions or in upstream regions. So this tells you that where the protein is bound, if it is bound in the upstream region just before the gene, it will have probably a regulatory function. And which genes that are actually bound, well, that's very interesting here, because we see that there are lots of genes that are involved in DNA regulation, so in transcription. We also find back a lot of genes that are involved in leaf development or in morphogenesis or cell differentiation. So this all links again back to the function that AN3 has in stimulating or in regulating growth, so the crosstalk between dividing cells and elongating cells. And apparently I talk too fast because I'm already at my conclusion. So I just showed you that I can use the GSyellow tag, which is a new kind of top tag, for analysis of plant proteins in either cell cultures, plants or corn, to identify localization patterns, interaction partners, or target genes. And I'd just like to thank everybody in my group. I have a wonderful group there, my PI, and yeah, all the technicians. So thank you for your attention.
Thank you very much. Questions?